All right, so Chapter 10, Part 3, we left off with Part 2 talking about photosystems. In this case, photosystems capture photons, pass the photons along to excite an electron, and that electron is then picked up by a primary electron acceptor. With that in mind, we're going to be looking at the two types of photosystems, photosystem 1 and photosystem 2. Photosystem 2 actually occurs before photosystem 1, and that's because photosystem 1 was discovered before photosystem 2. So 2 goes before 1, but the second one was discovered before the first one, hence why it's called photosystem 2, even though it occurs first. So to start with, photosystem 2 works best by absorbing wavelengths of light at 680 nanometers. The reaction center is of chlorophyll A is called P680. Photosystem 1 works best at 700 nanometers, and its reaction center is called P700. Now, photosystems 1 and 2, both part of the light reactions. Electrons, as we see, electrons flowing, kind of like the electron transport chain. The electrons can flow in one of two formats. They can either be cyclical or linear. Linear electron flow is the primary pathway, and it involves both photosystems and produces ATP, which we'll see coming out here, as well as NADPH using light. So to sum up this process, how it operates is a photon coming in from light hits a pigment and passes its energy among the molecules until it excites P680. This excites an electron in P680, and it bumps it up to the primary electron acceptor. P680 plus now forms as a result of loss of the negative electron, so this now has a positive charge, and is a very strong oxidizing agent. Water is split by enzymes. We see water being split over here. Water is being split by enzymes as a result of the loss of this. And the electron lost from the water split goes to restore P680 plus back down to a standard P680. As a result of the splitting of water, we have oxygen being formed off as a waste product. Lucky us. The primary electron acceptor here will carry an electron, so the electron essentially falls, as you can see, falls down an electron transport chain from the primary electron acceptor of photosystem 2 down to photosystem 1. Energy is released by this, which drives a proton gradient across the thylakoid membrane. These are embedded into the membranes of the thylakoid. So diffusion of these hydrogen protons across this membrane drives ATP synthesis, quite similar to the process that we saw in cellular respiration. Photosystem 1, like photosystem 2, transfers light energy, as we see light energy is coming down in here, getting bounced along to P700, causing another electron to become excited and picked up by another primary electron acceptor. As a result of the loss of the electron here, P700 has lost a negative particle, so it now has a positive charge. It's this loss of the electron giving this a positive charge that allows P700 to accept this incoming electron. So the electron over on photosystem 2 comes from water, but that same electron is what goes into P700 as these get excited by the photons and kind of dumped. These excited electrons from photosystem 1 are then passed down to a protein called ferredoxin, or FD for short. Once there, in ferredoxin, the electrons are then transferred to the NADP+, reducing NADP+ to NADPH. Once again, electrons are tied to hydrogens. the electrons of the NADPH then, in the H part once again, are available for the reactions in the Kelvin cycle or the dark reactions. And we can see that process going on off to the side here. The electrons going to ferredoxin, then to the NADP plus becoming NADPH, which will then be sent to the Kelvin cycle. And once again, we saw NADPH forming way back at the beginning of part 1 of chapter 10, actually. If you remember the NADPH going this way and the NADP plus going back, that's where this kind of falls into play. I'll actually go back to the image just to make sure that we're clear on it. So ultimately that NADPH is a result of photosystem 1, photosystem 1 and 2 are going on right here, forms NADPH, which goes over here to the Kelvin cycle and drops off the hydrogens, and now we have NADP plus going back. So let's take another look at the previous image, and here we are, and we can kind of very vaguely see that NADP plus going in and the NADPH going out. This was what we just looked at on that last slide. So what we just finished looking at was linear electron flow, once again because it starts on one side and it goes all the way down to the other side. But what about the other one? What about cyclic electron flow? Cyclic electron flow involves really just photosystem 1. The cyclic flow, just as the name suggests, is a cycle. We have an electron coming in, getting excited, going up to the primary electron acceptor, but instead of going off to ferredoxin this way to produce NADPH, it goes back this way by ferredoxin to the cytochrome complex to regenerate more ATP. So it's a process of pumping out ATP, and it just kind of continues around right in this stage. Some organisms, typically like bacteria, sulfur bacteria, have photosystem 1, but they do not have photosystem 2. This cyclic electron flow is actually thought to have evolved before the linear electron flow. And lastly, the cyclic electron flow may also help protect cells from light-induced damage. Okay, so at this point you might be thinking, well, you know, the photosynthesis sure seems a lot like cellular respiration. Well, it actually is quite similar. So you shouldn't feel like it is similar because, well, it is similar. Both chloroplasts and mitochondria are used to generate ATP, and they do this using chemiosmosis. Keep in mind that the source of energy is different. You know, from cellular respiration, the energy is coming from breaking apart glucose, whereas for plants it's from the intake of the photons from the sunlight. Looking at a third point down here, we actually can see that chemiosmosis occurs in both, and they're different, but there are similarities in how those processes occur. Couple more. Mitochondria, we have protons pumped to the inner membrane space. Once again, in cellular respiration, in the mitochondria, we have the electron transport chain embedded into the membrane of the mitochondria. And the same is similar with chloroplasts. The protons are pumped into the space as well to drive ATP synthesis as we have the diffusion going back in. Actually, both of these have that ATP synthase that you may recall from Chapter 9. We actually have that ATP synthase in this image right here. We can see that there's a membrane along with the electron transport chain. Regardless of the cycle, cellular respiration in the mitochondria or photosynthesis in the chloroplast, both the electron transport chain and ATP synthase are in the membrane. Keep in mind that mitochondria, we're talking the inner membrane, which is right in here. And the chloroplast, we're talking about the membrane of these thylakoids, so right here. Once again, we have the process of chemiosmosis going on, electrons being pumped up and then dropping going through a series of reactions which is driving hydrogen ions across the membrane, creating a gradient. And as the hydrogen ions build up in a gradient, they go back down through this protein ATP synthase. And as it goes through, this actually gives energy to ATP synthase to drive the production of ADP back into ATP with the addition of the hydrogen. So the electron transport chain generates not a whole lot of energy. It's really the ATP synthase. Keep in mind that this ATP, it's being generated by the light-dependent reactions or the light reactions. So this ATP is actually going to be going to the Calvin cycle, which occurs in the stroma. That being said, this is actually the inside of the thylakoid, and this is the fluid out here and out here and in here, the stroma, and that's where the Calvin cycle takes place. So we need the ATP to be on that side of this membrane. Looking at a little bit larger image of this, kind of once again expanding on it, trying to put it all together. Light going in the photosystem 2, exciting P680, electron goes up. We have a reduction of the electron transport chain driving down the hydrogen ions over the photosystem 1, exciting P700 with a little bit of light, exciting another one, electron that is, going to ferredoxin and out to NADP plus, forming NADPH. That's going out to the Calvin cycle. These input of the hydrogen ions drive the process of ADP transitioning into ATP by means of ATP synthase. Once again, this is outside in the stroma as well, and they're both going to the Calvin cycle. We're going to stop with part 3 right here. We're going to be moving into part 4. We're going to be looking a little bit more at the Calvin cycle and what the Calvin cycle uses this ATP and ADPH to perform.